Okay. Shall we start? Shall we start? Okay. Nice, nice. So, hi everyone. Glad to meet you both online and offline. This is our Chi-Hub seminar session 13, and today we will have two talks from Melody and Avalon. So I think I'm gonna introduce you both at the beginning, and when you finish the talk and one by one, then we will have the questions, QA sessions at the end. So for the talk one from Melody, he will talk about the title about From Eye to Brain, Investigating EEG and the Retinal Measure for Early Detection of Alzheimer's Disease. So this presentation will explore how retinal imaging and EEG-based biomarkers may support early identification of Alzheimer's disease. Melody will discuss the current findings, methodological considerations, and the potential for integrating multimodal physiological signals in future diagnosis pathways. So that's for our talk one. And we also have amazing talk to Avalon. He will talk on the title of Granular Changes in Brain Networks, Multiplex Nodal Modularity for Alzheimer's Disease Characterization and Simulation. So Avalon presents new work on multiplex nodal modularity and its role in understanding structure and functional brain network changes in Alzheimer's disease. And the talk will highlight how network level modeling can support disease characterization and simulation. So the stage is yours, guys, without further ado. And yeah, I'm looking forward to it.
Okay, that works.
Yeah.
Okay.
So probably need to speak up a little bit louder, so something with the mic as well. So, but anyway, yeah.
Thank you everyone for having me here today. My name is Melody Sequeira. I'm a second year PhD student in the Translational Neuroscience program here at the University of Edinburgh. And today I'll be presenting my PhD project and some of the findings I have from my first year doing this project, where I look at accessible and non-invasive tools, particularly those that measure changes within our eye, looking at the retina, and looking at brain function using EEG for early detection of Alzheimer's disease. So I always like starting my presentations with this slide, which are quotes of people living with dementia or loved ones and carers of people living with dementia that kind of talk about their journey through diagnosis. And through these quotes, it's very evident that diagnosis takes really long in dementia. And if you look at some of these quotes up here as well, they talk about the uncertainty of not knowing what they were feeling and why they're feeling the way they do more than the diagnosis itself. And so it remains extremely important that dementia does not go unseen. And that, you know, some action has to be taken to make diagnosis more accessible to everyone. And so with the, I guess, emotional burden that comes with not knowing, or the uncertainty of not knowing what you're feeling the way, why you feel the way you do, 1/3 of the people living in the UK do not have a diagnosis in dementia and kind of go through this emotional burden. But more than anything, there's also this economic burden of dementia that is predicted to double by 2040. And so at this stage in time, it remains absolutely crucial that these early and accessible diagnostic tools for dementia diagnosis. So over the last few years, retinal imaging has gained a lot of interest in being a possible accessible avenue to detecting Alzheimer's disease early. And as you can see from some of these, you know, headlines in articles, blog posts, and a lot of research that's been happening, the eye can possibly tell us changes that happen in our brain, possibly earlier before we see it in brain imaging as well. But why would that be the case? Well, the eye is widely considered a window to the brain, and a lot of blood vessels within our brain extend onto the eyes as well. And with the rising interest in retinal imaging, there's been a lot of studies that look at the brain and eye interaction, especially in the context of Alzheimer's disease, where a difference in the thickness of these retinal layers, especially the macular and the retinal nerve fiber layer here, show association with differences within the structure of the brain. So with differences in lower brain volumes, especially in the occipital and the hippocampal regions. But when we consider a condition that is defined by cognitive impairment, it also remains important to look at brain function and cognition. And how can we do that in an accessible way? Well, EEGs could be a possible solution to do that. So electroencephalograms, or EEGs, are a cost-effective, accessible tool where you attach a cap with electrodes to your scalp that read real-time electrical activity happening in your brain. Now, this can happen when you're at rest or doing a task such as a memory task. And these are quite nice in understanding real-time brain activity that give us readouts of brain function. They are non-invasive modality. They are quite accessible and cost-effective compared to some of the other modalities out there like functional MRIs. And they can help us detect subtle changes using computational modeling. So combining the two modalities together, my PhD project, or the big question I'm trying to answer through my PhD, is to what extent can these non-invasive retinal and brain modalities detect and reflect changes related to Alzheimer's disease? So to do that, I'm first looking at the retina. So I'm looking at particularly the back of the eye, which is the retina. And this right here is called a fundus photography. So this is an image that you'd get when you go for an eye test. It is free of cost here in Scotland. And through these images, actually, we can get really good insights on the structure of the retina, how this might change through the disease as well. So some of the things we look at are the complexity of these artery and vein networks, looking at how it branches through the retina. We also look at changes in the curvature of these arteries and veins and how they change over time. Something that is of interest as well is the optic disc itself, and if it turns paler over time, and if that could be a sign of disease within the brain. I also look at EEGs, and with EEGs, I look at how different frequency bands and the power that they have through different conditions. So during resting state, during task phase, I look at how different brain regions interact with each other functionally using functional connectivity. And I also look at how complex these brain signals are in individuals using complexity measures. And I look for these patterns in three different groups of people. So the first one are individuals at risk that have family history or the APOE4 gene, which is quite influential in Alzheimer's disease. I then look at individuals that have mild cognitive impairment, which is this transition state between healthy aging and dementia, where about 10 to 15% of this population turn to having Alzheimer's disease. But this particular condition is quite interesting because it's quite heterogeneous in its nature, where some people actually stay stable with mild cognitive impairment through their lives, and some actually go back to being healthy. So this condition on its own is quite heterogeneous and raises a lot of, challenges when we look at changes within our brain and changes within the eye as well. And finally, I'm hoping to look at individuals that have early Alzheimer's disease as well. And so the idea is to look at the progression of the eye and brain measures through the disease course. So in today's talk, I'll be talking about the retina and EEG independent of each other, and I've run these analyses on different groups of different cohorts. So the first one is retinal analysis in individuals at risk of Alzheimer's disease. And this was done in a Prevent Dementia cohort, which is this really good study. It's multicentric all around the UK. And it aims to identify early signs of dementia in midlife. So the age range of these individuals are 40 to 59 years. And the aim of our study within this project was to evaluate changes within the retina in individuals that have heritable risk factors. So the APOE4 gene or family history. And this was done by dividing the group into three categories: low risk, which was people that didn't have any risk factors; medium risk, which is they had either one of the two risk factors; and high risk is that they had both risk factors. And this was done within the Prevent Dementia cohort. So within this data set, I had two different modalities of retinal imaging. Fundus, which I introduced previously, is very accessible. You get it through an optometrist. Ultra-wide field imaging is now apparently available in booths for an additional £50 charge. But what ultra-wide field imaging does is it helps us look at a wider view of the retina, helping us also look at changes within the periphery, which has been noted to change in Alzheimer's disease, where the vessels in the periphery get thinner over time. So overall, the data was about 100 people in both modalities. Their age was about 51 years, and they were predominantly female. So before I get into my results, I wanted to show you how we go about analyzing these images. So this is what an ultra-wide field image looks like when we capture it and project it and everything. Our next step is to QC these images to see to assess the quality of the image. And this is because image quality has a huge impact on the features we then derive. So what we look for here are how much of the eyelashes cover the arteries and veins, what the signal quality is, the illumination, the darkness of the image. Can we see every vessel, artery, vein? Can we see the optic disc clearly, or is it blurry? So kind of depending on those parameters, we decide if they are pass or borderline, which then go on to the next stage of feature extraction, or we reject the image, which is excluded from the study at that point. After that, we segment the image, and this segmentation is done using a deep learning model. But to train this model, we had to segment these images by hand. So each artery and vein is segmented. And we try to be as precise as we can with the segmentations, especially getting into the smaller, like microvasculature here around the fovea. And then the segmentation pipeline segments these arteries and veins, and then we get metrics for arteries, veins, and then vessels together. So once the segmentation is done, we run these metrics or these images through two softwares. The first one is Ultralyzer, which analyzes the ultra-wide field images, and Automorphalizer, which analyzes the fundus images. And as you can just see from the numbers, you get a whole range of features, like 46 features for one image, and 49 for fundus. So it's a lot of metrics to get your head around. And so what I wanted to see with this project was what features actually capture the largest variance in my images, and what can help me understand this better and in less features. So to do that, I did a principal component analysis to identify which features explain the most variance, and then looked at the top 50% of each principal component analysis to see which feature contributes to that principal component. And so doing all of this, I got 3 principal components for fundus images. The first one is local caliber, looking at the width of each vessel in the retina. The second one is vessel complexity, that is measured by vessel density and fractal dimension, which looks at how these arteries and veins branch through the retina, and also how much they cover the retina itself. And then tortuosity, which from this image, you can see the curvature in the vessels, and that is what we measured through tortuosity. So analyzing the fundus images, this is what we find. So from first glance, you might be able to notice that there's actually changes within our eye as well. So left and right eye show different measures, where right on average shows as higher measures compared to left. And I think this is extremely important when we look at the retina as well, and to compare both or include both eyes in our analyses. And while I ran linear mixed effects models on these metrics, fractal dimension and vessel density that give us the understanding of vessel complexity actually flag up as being significantly different in high and medium risk group compared to low risk group. And what we actually see is different from what's established in literature, where there is an increased vessel complexity in the high and medium risk group compared to low risk group. And this is quite interesting because at-risk individuals are quite underexplored in retinal literature. And so it could just be that this is compensatory mechanisms happening earlier on in the aging stage, and could later lead to decreasing vessel complexity, which is what is noticed in literature. But this is something that I have to look more for, and I hope to do this through the longitudinal analysis I'll be running next. Something quite interesting with these features is that age has a huge effect on these metrics, and so does image quality. And I think this is extremely important to consider, especially when we think about converting this technology to community settings where people from wide ranges of age will come, and you'll get wide range of image quality as well. So then moving on to ultra wide field images, very similar to fundus, we get the same kind of principal components. So principal components defining vessel complexity and tortuosity remain the same. We also get optic disc measures, so difference in the diameter and area of the optic disc itself was flagged up as being variable across the images. And width metrics, which is another quite interesting one, which looks at how the vessel widths change as you go from the optic disc to the periphery. And this is quite a good metric, especially for ultra-wide field imaging, because it takes advantage of the peripheral view that we have in these images. And so after doing that, we get 6 metrics from these 4 principal component analysis. And once again, running linear mixed effects, we see vessel density as high in high and medium risk group compared to low risk group. But quite interestingly, vessel width also increases in people with high risk compared to low risk. And so does the vessel width intercept, which is the vessel width at the optic disc. And so again, it quite aligns well with our fundus image analysis, where we see increased vessel widths that could be signs of compensation, but in small vessel disease actually have been reported to be associated with changes to small vessels themselves that leads to reduced blood flow and inflammation. And yeah, so that could be one possible explanation as to why we see increased vessel widths. So moving on to my EEG analysis, I did EEG analysis in individuals with mild cognitive impairment. And so to do that, well, firstly, a huge thank you to our collaborator, Mario Parra, who's based at the University of Strathclyde, because of whom we got access to this really good data set. So the aim of the study was to assess differences in brain activity and task dynamics between individuals with mild cognitive impairment and those that are healthy using graph-based synchrony. So the sample size is quite good in this, where we had 107 controls against 108 MCI individuals with ages about 60 years old, again, predominantly female population. And the task that they did here was called a visual short-term memory binding task that is quite sensitive to early changes due to Alzheimer's disease, where individuals are first shown 3 shapes with different colors. And this is the phase that we call encoding, where they're kind of looking at the objects and trying to understand what they're seeing. And after a time delay, they're shown 3 shapes with objects with colors again, and are asked if this was the same as the one they previously saw or different. And that is what we call the recall stage. So we acquired EEG data off of this task, and then we convert them into graphs, which Avalon very kindly did for me. So these graphs are defined by phase-like index, where we look at how ahead or behind one signal is of the other. And it looks to see how one brain region, which is a node, is how we define it in our graph, connects to another brain region using brain functional connectivity metrics here. And the edge here is defined by phase-like index. And using that graph, I computed measures of synchrony, which basically tells us how together or how synchronized the brain activity was when participants were doing the different phases of this task. So overall, what we found was that there was higher synchronization, so there was higher activity in MCI individuals compared to healthy controls. And this was consistently seen in the theta, alpha, beta, and broadband. And what's mostly of interest is the theta and alpha band, because they're like extremely important and have been well documented in Alzheimer's disease. These are bands that kind of look at cognitive functioning in resting state, but also in task-based. And we see the same pattern where MCI have higher synchrony during recall compared to healthy controls. But what's more interesting is when you look at the brain region activity itself, the regions that flag up that are highly synchronized are the ones at the back. So the occipital region, the hippocampus, the parietal region, all of these that play a huge role in the binding network for visual memory. And these were flagged up as being highly synchronized compared to healthy controls, which I think is quite evident through these images here. And so, And so what we can infer from this is that this activity of hypersynchronization during recall is suggestive of increased activity that comes with greater cognitive load. So recall requires a lot more cognition than maybe looking at objects on the screen. And so this increased cognitive load could be a possible reason why we see increased activity, where the MCI are just trying to compensate for a lot of that cognitive load. And so overall conclusions from the sub-studies I did in the eye and the brain, in the eye, we see that people with heritable risk factors have increased vessel complexity and vessel widths. It could be understood as compensatory mechanisms or something to do with changes within the structure of the brain. Within individuals with MCI and EEG, we again see a similar pattern of compensatory mechanisms with higher activation during increased cognitive load. And so the next steps of my PhD project is to look at how these brain functions change in individuals at risk of Alzheimer's disease. Do we possibly see signs of compensation as early as, even before they're diagnosed with any condition? Or our genetic risk factors have an influence in our brain function? I then hope to look at changes within the retina in individuals that have a diagnosis, so that are diagnosed with NCI or Alzheimer's disease. And then I hope to establish kind of this mechanistic association between changes in the eye and changes in the brain. And if, in fact, changes in the eye could tell us anything about changes within our brain function and go beyond kind of brain structure changes. And this work would not have been possible without the great team that supports me through this all. A huge thank you to my program, the Wellcome Trust Translational Neuroscience Program, that's funding this project. A huge thank you to my supervisory team, Dr. Javier Escudero, Dr. Tom McGilvray, Dr. Emily Adrian, and Professor Savanka Pal, who have guided me through this project. A huge thank you to our collaborator as well, Professor Mario Parra, and huge thank you to both my labs, the Biomedical Signal Processing Lab that's based here in the Chai Hub, and Velvet Curl Ophthalmology Suite based in the IRR SAP. Yeah, thank you.
So if there is no like urgent questions pop right now, I don't see any urgent questions on chat. Shall we move to Avalon's talk? And we can have like a question collected at the end, and then we can ask all together. Is that okay? Yeah. Yeah, nice. Okay,
I'm gonna have to share my whole screen because it's not popping up.
Yeah, we can see that.
That's should be okay.
Yeah.
Okay. Perfect.
Yeah, perfect.
Let me just move this over here
Yeah.
in case.
Yeah.
All right. Well, thank you everyone for coming. My name is Avalon Campbell Cousins. I recently finished my PhD with the School of Engineering. I'm in the same group as Melody, who just presented, and I am going to share with you some of my PhD research, specifically around how we can utilize granular brain network measures in this sort of multiplex model that I'll describe in quite shortly. And how this is really important for Alzheimer's disease characterization and simulation. I was a bit ambitious when giving the title to the Chai Hub. Given time, I will only briefly talk about the simulation portion of this talk, but of course I'll take any questions on it afterwards. Oops, sorry there. So as Melody did an amazing introduction on Alzheimer's disease, pointing out how much of a burden it is emotionally and financially, not just in the UK, but across the world, with this projected to only increase as time goes on and people live longer lives. And understanding the early stages of Alzheimer's disease is so important because it allows us to give, as treatment becomes available and develops, we can provide it at an earlier stage before significant damage has been done. And I'd like to start this talk by giving talking about the brain as this sort of complex system, not only along sort of space, but time, and also how these different things interact with each other. So not just the global architecture of the brain, but also sort of the local connectivity and the relationships between these sorts of brain regions. And how they evolve in time, in short time scales, like say a 10-minute session, or across sort of stages of Alzheimer's disease. And this can be captured in a variety of ways. And in the research I'm going to be talking about today, I'll be talking on functional MRI that images the blood movement in the brain. And as your brain regions are very hungry and active, they require lots of blood movement to support brain function. And sort of the white matter highways imaged with diffusion tensor imaging that connect them and support the sort of structure of brain communication. And we can model this as a network, and Melody touched on how we can sort of, at its most basic, you can look at nodes and some relationship between them. Think of this as two people, and perhaps they are connected via a friendship or a parent-child relationship. These sorts of things in a social network are quite common, but can be easily described in the brain as well, where individual brain regions can be described as nodes. And the connection between them can be some relationship. In the case of functional MRI, you might want to look at the correlation between blood movement between, say, two brain regions. And when this relationship is quite strong, you might say that these two brain regions are co-activating with one another, which is quite important when analyzing, say, task-based dynamics in brain networks, where in a motor task or a memory task, you have multiple functional systems working together to promote brain function. And in the case of DTI, these connections can be a direct sort of structural relationship based on the white matter density between two regions. Or in the case of my research, we looked at the fractional anisotropy, which is a measure of brain white matter microstructure. And so the motivation for using network-based models for diseases such as Alzheimer's disease is that we know that as Alzheimer's progresses, it causes massive structural and functional organizational changes in the brain. As disease and damage occurs, brain regions tend to start to communicate less so brain-wide, but more so functionally segregated. And this has downstream effects of especially in Alzheimer's disease, memory loss and impairment. And this motivates the development of novel network measures that can be sensitive to these early stages of the disease. But one limitation of classical networks is that they're often looked at in quite a static nature. You have a 10-minute, say, fMRI scan, and you have one network describing all of the complex dynamics that are occurring, which means that you can miss out on some higher order interactions that could be occurring. And that's where multiplex networks come into play. Because with a multiplex network, we can construct from our, say, fMRI time series, individual windows, which will be our networks. And these can be, are really nice when looking at task-based data, because each of these time windows can be a stage of your task. And you can track those dynamics across time when you model them as a network with interdependencies that connect across time. And multiplex network is just a type of multi-layer network, which lends itself well to network neuroscience, specifically because each of these will be a brain region, and your brain regions are changing over time. You know, they're well, on long enough timescales, yes, but in the short term, at least, no. And so you can track sort of these connections across time. And so one of those network measures that's particularly well suited for looking at organizational-based changes that would be occurring due to Alzheimer's disease is modularity. And modularity, specifically the Louvain method, which we're exploring, which is widely used not just across for brain network-based studies, but also with other biological networks. Its goal is to segregate the brain as well as it can into distinct communities that are more working together, more so within their own communities than they are between different communities. This has been extended to multiplex networks. So now our communities don't just exist within one window of time, but across time. So you have these sort of communities that are across your different time windows that would be lost if you were to look at your entire fMRI scan as one network. But very briefly, before I go on to the next slide, one of the limitations to modularity and how it's been used in the literature in both single and multiplex networks is that we have a global measure of modularity that says how segregated our network is into these different communities. And we also have community assignments, which is like a label that we're giving to each of the brain regions in our network, saying they're working in one group and these are working in another. But we don't know to what extent an individual region of the brain contributes to this overall global network architecture. And so a contribution that we've made is nodal modularity, which is a novel extension of classical modularity extended to individual nodes, where now you can track how nodal modularity changes as time progresses, Here's a shameless plug of the research article if you want to know more about the mathematics behind it and these sorts of things. And it's been developed for both classical single layer networks and multiplex models. And this gives us a more granular view of what's happening when reorganization occurs due to Alzheimer's disease, and what sort of brain regions we're seeing these changes pop up in. And so we looked at this in a neuroimaging cohort of a pretty small one, I must admit. But the important thing to note is that we have healthy controls, people with early mild cognitive impairment, and then 6 of our subjects after a 2-year follow-up progressed to Alzheimer's disease. So we have these MCI converters, MCI, early MCI healthy control. And for each of these subjects as well, we have DTI measuring that white matter structure, as I mentioned, and fMRI for 2 specific tasks. It's the same visual short-term memory binding task that Melody introduced earlier. But we're also looking at shapes on their own and colored shapes as 2 separate tasks. So it's a small data set, but we can probe a ton of different hypotheses based on the task, based on different network structural, or different networks that are based on structural brain networks and functional brain networks. So very briefly, how this is modeled, we take the visual short-term memory binding task, we break it up into 2 time windows because it's a very fast task. And so the temporal dynamics are so quick that we ended up having to combine the encoding and maintenance phase into 1 time window. And then the probe phase, which is that sort of recall step where they have to answer whether or not the colored shapes or the non-colored shapes have changed. We put this into a 2-layer network, a multiplex network, and we can sort of track when we're calculating nodal modularity how that changes in the 2 separate time windows that represent the 2 phases of our task. And what are the results? Well, we did statistical analyses with permutation testing. These have been also FDR corrected, and we did receiver operating characteristic as well. We took sort of this quite conservative approach to our statistics because we're so limited in the number of subjects we have. But what was most striking is that we didn't see too many differences in when we were comparing controls versus early mild cognitive impairment and mild cognitive impairment that didn't convert to Alzheimer's disease. But when we look at our mild cognitive impairment that converted to Alzheimer's disease compared to our controls, note that all the labeled regions here are those that pass the statistical thresholds I just mentioned. We see massive changes. And this is really encouraging because it suggests that perhaps nodal modularity could be a way to distinguish between those who will progress to Alzheimer's disease in sort of this 2-year follow-up, but maybe in further research, we could extend that to sort of longer scale predictive case. And due to time, I have to sort of just rush through the DTI results. But what was encouraging too is that we saw that our hypotheses related to multiplex modeling giving us deeper insight into the sort of higher order interactions was the case. So we saw that for our multiplex models that the statistical results were much stronger than if we were to look at each individual layer separately. We saw that the locations that we found that showed abnormalities in nodal modularity and fMRI were common locations for amyloid beta deposition in poor memory binders specifically, and similar results in DTI, and also really importantly that the shape task, which our collaborators found was not a good indication of early MCI when they were looking at the shape task specifically that weren't colored shapes, they found very little difference between people with mild cognitive impairment and healthy controls in terms of their performance. But the difference was massive for the binding task. We saw that directly reflected in our fMRI results, where we found no results for the shape task, but we found large number of abnormalities for the binding task, and a lot of them existing in, say, visual occipital systems where we would expect them to. And then we did a bunch of other stuff with nodal modularity that I can't get into today, but I'll very briefly run over it. We introduced this measure of collapsibility, so it allows us to look at the variability of nodal modularity across sort of multiple layers of the network. The importance of this is that if you have a system with hundreds of network layers, having nodal measures for every single one of them is very difficult to interpret, but looking at the variability across the network is much easier. Nodal modularity and collapsibility have been verified across a number of surrogate network analyses that we did. We applied it to the EEG frequency, EEG networks that Melody presented earlier and found some interesting results as well. They're not as interesting as the fMRI results, just because those were earlier stages of MCI. And there's this very interesting sort of early stage research that we did around targeted attacks. So the idea being that you attack a network in some way when they're healthy, and that simulates the network that you expect them to develop to as they progress into Alzheimer's disease. And previous research had looked at attacking these based on sort of degree-based node hubs, and we thought that nodal modularity might be a better targeting mechanism, and early research does suggest that. And so some limitations and future work to consider. Of course, the sample size we worked with was very small, and so further research looking at nodal modularity and collapsibility in much larger cohorts to verify this behavior that we see in terms of predicting those who will go on to progress to Alzheimer's disease from mild cognitive impairment. And another big area of research I think that would be quite interesting is using nodal modularity with other clustering algorithms that are faster or have less sort of degenerate solutions than the Louvain-based methods have. And the other thing is that I think simulation-based models could be very interesting to look into for AD subtyping. A lot of recent research is suggesting that there are many different trajectories that can occur within Alzheimer's disease. And so if you had a method of simulating sort of attacks to these networks in different ways, having separate ones for each trajectory pathway, then that might be quite an interesting way to probe these different AD subtypes in the future. And so lastly, as a general overview, we looked at the brain as a sort of complex system where not only are there interactions within the brain, but across time. And we want to capture some of these higher order levels of interactions because Alzheimer's disease is so complex as it is, and to see if these higher order interactions will help us to understand the disease better and even predict its conversion to Alzheimer's disease. And we introduced this novel measure of nodal modularity, which is an extension of classical modularity, to measure how brain organization at a granular level changes across disease. And sort of the combination of these two ideas led to, the data suggests that nodal modularity could characterize a key turning point of Alzheimer's disease, which is where people with mild cognitive impairment go on to progress to further stages. And lastly, I want to thank both my research group on the left here, which is the biomedical signal processing group that shares an office with the Chai Hub, and also my collaborators, Mario Parra, Federico Guazzo, and Mark Baston, who I work with very closely, along with contributions from NHS Scotland for the data collection in doing this fMRI and DTI study. And yeah, if you have any questions, feel free to email me. The code for nodal modularity and collapsibility is available on my GitHub, so feel free to play around with that. And yeah, I guess that's all for me. Thank you.
Okay, wonderful. Any questions offline, online?
I think there's a question in the chat from Ahmed.
Yes.
Wada.
Ahmed, would you like to open your mic and you can ask directly?
Ahmed? Should I just read out loud? This is a question for Melody. Considering that he said, Ahmed said, hello, nice work. Considering that some known retinal diseases, such as diabetic retinopathy, which is a vascular disease, are associated with dementia, how did you tackle that? And did you use only normal, in brackets, healthy fundoscopy images?
Yeah,
Melody,
so,
this question is in the chat if you want to read out.
So I think the question is really valid, and it's kind of a challenge we face in retinal imaging, because with diseases like diabetic retinopathy, they directly affect the changes within your retina. So we exclude these participants. So any disease that affects the retina or any ocular disease is rejected. They're all excluded from our study. And then if we find any images that are low quality, sometimes due to cataracts and things like that, they're also excluded from our analyses. The data that I presented, they're all healthy people. And yeah, they don't have any diagnosis or anything.
Good.
Thank you, Melody.
I also have a question to Avalon as well. So all the networks you present, including the nodal networks and within like their communities, the hierarchy, is this all these like a community node type all predefined, and you just map with the images?
So if I'm understanding your question correctly, so just as a quick overview, we have the brain regions, so the nodes in our network are predefined.
Yeah.
They're based on an atlas, which is a segmentation method for the MRI images that give you, say, the amygdala, the hippocampus, like all the different brain regions. We have 85 of them in our case. And the connections between them are taken directly from measurements from the images as well. So that's sort of the basis of our network. From there, you have to maximize your modularity quality function. So it's you're running this iterative process that further tests, okay, if we have these and these groupings, are they is this function maximized or not? And you keep going until the segmentation, or the segmentation, not the image-based segmentation, the segmentation of the nodes into groups. That labeling is sort of maximized. And then from there, you calculate the nodal contributions on that network. Did that answer your question? Okay, perfect.
I had a question for the node modularity that you're doing. Does that mean that you highlight the nodes that you consider most important?
So in this case, it's just a measure between 0 and 1. So you have across your entire network, you have all of these different measures of nodal modularity. And then we're comparing those from healthy to disease. So you have the whole cohort and you're doing a statistical analysis between.
And then one would be high activity versus 0 being no.
It would be high contribution to overall. I mean, if it was, it would likely not get close to 1. Usually these are really small values. So in the classical modularity sense, if your network had a modularity of 1, it means there's these very distinct communities. Maybe there's a highway between them, where they're really densely communicated and there's like one connection. But in the brain, it's much more complex. You have connection. Where in functional MRI, you have almost a fully connected network, just where edge weights can be very, very close to 0, when there's almost no association between the blood movement in those brain regions. And so this measure of nodal modularity is how much is that brain region contributing to network structure. So for instance, if you thought of it like your PI would have a really high nodal modularity in your group, because he's connected to all of you and he's driving like a lot of the discussions, and he has connections to collaborators and to and farther reaching connections to government and all of these different things, right? And so his value of nodal modularity would be very high in that sense. So you can think of it more, it's more closely akin to in like social networks, they talk a lot about like node hubs and people who are very influential in a network. And in the brain, it's the same. It's like nodal modularity is another indication of how important that brain region is to overarching brain structure. Yeah.
Thank you.
Any other questions from online audience or? So no more, then I think we can call it today. Thank you, Avalon and the Melody, for presenting very interesting works, and I think it's very important to understanding of the brains and the photos images, EEG signals released to the dementia diagnosis. And I hope you all enjoyed this seminar today.
Yeah, I really enjoyed it, to be fair. It was brilliant. Thank you ever so much, both of you. And I find it really fascinating just to see what you're working on. It was eye-opening. Thank you both.
Okay, so
Thank you.
Yeah, I think we can call it today. And thank you for participating offline and online. And I hope you have a nice day. All right, I'll see you next seminar series.
Thank you all. Bye.
Thank you all. Bye.